Hello, Professor and class, and good morning or afternoon or evening. My name is Luke Schwagler from Ashland, Montana. This is my microcosm project for General Ecology EVSP 416. So the first thing we need to do is identify what exactly a microcosm is. To me, they are tiny little ecosystems in a jar. They allow us to control different variables that might affect how the ecology in that tiny system reacts. We can control the relationships between organisms. We can control things like sunlight or oxygen, gas exchanges inside the jar, depending on what we do with the experiment. These things have been used in almost every facet of ecology. They allow us to kind of study on a small scale what might be happening, where you collected these organisms from in the environment, and show us what we might be doing to affect how that ecosystem is functioning. I hope to get a better understanding of broader ecological processes using this method and model to really show what human effect has on these processes. And we could apply that to a broad natural ecosystem, and it may help with some decision making for some type of development or something like that, or whether or not to use certain things such as chemicals in an area and what the effects might be to that ecosystem. These things also show us how organisms interact with one another depending on what we do to the environment. They show competitive abilities and the lack thereof of organisms, and which ones may benefit from changes to their environment or which ones may suffer from changes to their environment. So the hypothesis I came up with was oxygen levels in the microcosm control life through gas exchange processes. Therefore, any aerobic organisms will perish. Anaerobic organisms will survive due to less competition from aerobic organisms in a suitable low oxygen environment. So for my microcosm project, I used two separate jars. Jar 1 was approximately 355 cubic centimeters in volume. Jar 2 was approximately 927 cubic centimeters in volume. Jar 2 was approximately 40% bigger than Jar 1. I added 64 grams of clay and decomposed organic matter to Jar 1 with 237 milliliters of water and 14 grams of surface algae. Jar 2, I added 128 grams of clay with decomposed organic matter, 474 milliliters of water, and approximately 14 grams of surface algae. And I also added one medium-sized eggshell cracked in half and 6 inches of cattail to Jar 2. The thinking behind this was that Jar 2 would have more resources than Jar 1, and the organisms within might benefit from that. The samples came from Black's Pond near my hometown of Ashland, Montana, on the Custer-Gallatin National Forest where I currently work. Black's Pond is fed by Cow Creek Spring. This is a perennial spring coming out of shale bedrock with distilled quality water. The microcosm will run from August 5th to September 16th, which is 43 days in total. For week 1, I placed it outside on a picnic table, but due to weather concerns, as this time of year we typically see thunderstorms that can carry large hail, glass is the jars being glass, I was afraid that they might be damaged, so I slightly moved them to a location just right next to it that's protected from the elements. They would receive the same sunlight and they're at the same elevation as the previous location. The samples would receive approximately 14.5 hours of sunlight. This would decrease as the days get shorter, approximately to 13.5 hours as we progress through the project. The temperatures here have been in the low 40s to all the way up to 110 degrees Fahrenheit. We see pretty big swings in weather here in southeastern Montana. The photo here is the exact location that I collected the samples from in that little gap between the cattails that you see there. So this slide shows week 1 and 2 of the project. I collected the project on Wednesday of the first week and gave it time to settle. Took the first picture on Friday. You can see in week 1 right here that there were a few microorganisms swimming around in jar 1. The algae you can't see because it ended up sinking to the bottom when I placed it in here. This is this mass here, and here was the algae that I placed in. You can see jar 2, week 1, the cattails are still green and eggshell is still vibrant white. As we progress to week 2, the water got murkier and the microorganisms inside of the jars reduced significantly, nearly to where there was maybe 2 or 3 swimming around in there. You can still see the algae at the bottom on both of them. The organic matter in week 2 did get quite a bit darker in jar 2. Cattails still remained fairly green with slight orange coloring towards the tips, and the eggshell remained white. As we progress to weeks 3 and 4, week 3, jar 1, there was really no noticeable changes other than the organisms in the jar that was swimming around totally died and sunk to the bottom. Jar 2, you can see that the cattails are more of a yellowish color, and so is the eggshell, potentially decomposing. As we move to week 4, again, jar 1, slight changes. The water may have gotten a little more murky. Jar 2 in week 4, gas exchange started and bubbles were rising to the surface. These are the only real noticeable changes from weeks 3 and 4. Week 5 and 6, you can see week 5, jar 1 has bubbles forming at the surface, gas exchange occurring there. Jar 2, gas exchange seemed to have slowed significantly. A lot less bubbles this week. The cattails, you can see here, are more orangish brown color as they decompose. Jar 1, week 6, gas exchange is still taking place, bubbles on the surface here. Jar 2, this section here is algae on the surface. Gas exchange still taking place. As you can see on the surface, bubbles were still forming there. The cattails decomposed significantly in jar 6, in week 6, on jar 2. Week 7, the final week of the project. You can see in jar 1 here, there are significantly more bubbles at the surface. Gas exchange taking place. The water in jar 1 is more of a yellow tinge, and it appears that growth may be occurring towards the bottom of the jar on top of the sediment levels. Jar 2, you can see the algae here has grown a little bit, and it would appear that some type of water-loving plant at the bottom is growing. Water in jar 2 also a more yellow tinge to it, and the cattails are nearly completely decomposed. Some of them started sinking to the bottom. The major findings and take-home messages and what happened in the Microcosm project. It was a lot slower process than I had expected. The additional resources in jar 2 didn't seem to create that big of a difference. As far as the observation supporting my hypothesis, I would say that yes, that the aerobic organisms perished while the anaerobic organisms survived. This was due to the low oxygen environment that the project was conducted in. You could see that there was some growth of anaerobic organisms like the algae and the plant growth at the bottom of the water level on top of the sediment. As far as any microorganisms surviving, none that I could see. This was probably due to the lack of oxygen within the jars. The first concept that was clearly evident in this project was abiotic limits, or limits set by resource availability such as food, water, oxygen, and sunlight. To build off that, the law of minimum was also evident. The law of minimum, or Liebig's law, states that growth is dictated not by total resources available, but by the scarcest resource. We saw in this project that oxygen was the scarcest resource, and this heavily controlled growth rates within the ecosystem. The law of tolerance was also clearly evident in this project. The law of tolerance, or Shelford's law, states that an organism's success is based on a complex set of conditions, not just the most scarce resource. This was evident as some aerobic organisms were able to survive to a point. Once they reached their level of tolerance that they could no longer tolerate, they perished. The second concept that was clearly evident was biodiversity. This experiment lacked biodiversity, which led to the death of many organisms within the experiment. When you take a ecosystem with large amount of biodiversity, such as the pond that this was collected from, and you condense it to a small system with limited oxygen, you greatly decrease the biodiversity and reduce the relationships those organisms may have had with other organisms in that larger ecosystem. This can lead to a decline in a stable system. Say that these organisms have relationships with thousands or millions of other organisms within the system. If you take those relationships away, that system starts to decline. On the flip side of that, some of these relationships may be negative relationships, or they may have interspecific competition between organisms. So moving those organisms to an area with less competition could potentially benefit those organisms. In this case, it didn't seem to have that benefit, as most organisms slowly grew or failed to grow at all. The third concept that was clearly evident in this project were plant adaptations. We could see that algae has adaptations to survive in low oxygen environments. These anaerobic conditions benefit the algae and negatively affect the aerobic organisms that are in this anaerobic environment. So further research, or a hypothesis that I could test, would be oxygen levels in the microcosm control life through gas exchange processes. Therefore, any aerobic organisms will survive. Semi-anaerobic organisms will survive due to less aeration from previous environment and a suitable oxygen environment. So to do this, I would expose the microcosm to more oxygen, say leave the lid of the jar off. This would allow more oxygen to enter the environment and could potentially benefit the aerobic organisms within the microcosm. With where I collected this sample, the water is constantly moving, though it didn't seem like that from the pond. It's slow moving. This algae is able to survive along the shore of the pond where the slower moving water is at. If you left the jar off the lid and still had the algae in there, you would have less aeration due to less moving water. So potentially just leaving the lid off the jar could increase the survivability of both aerobic and anaerobic organisms within the system. To improve this project, I would like to increase the size of the microcosm. From our readings, I found there are large microcosm projects out there that can be, say, the size of a football field. Not saying that I would want to do something like that, but increasing the size of the microcosm would just allow for more biodiversity within that system and allow for more relationships to occur between organisms, and you may see more positive and negative relationships, or more competition or less competition within that. I would also, if I had to do it over in the same jars, I would decrease the water within the jars. I don't feel that I left enough space in the jar for a lot of gas exchange to occur. I think that's what ultimately led to such slow growth and the death of some organisms. And I would like to add more organisms to the experiment if I could, maybe get different types of plants that may survive in different types of conditions. That way you could see plant adaptations. You could also see levels of tolerance of different types of organisms within the system. So as far as microcosms and their relevance to general ecology, they can help us predict how ecosystems will react to ongoing changes within the environment that they're currently in, and it can also tell us what specific conditions are required to make that ecosystem a more stable environment. These Microcosms can also provide us with important information on ecological processes. They can show us what one little tweak here or there can do to an ecological process that may be detrimental to that ecosystem. And I think the biggest thing that these models show us is what human interactions or disturbances can do to ecological processes. These kind of give us the opportunity to impose those disturbances on a small scale and look at the effects that they may have on an ecosystem, whether it be a positive effect or a negative effect. It shows us what our actions will ultimately do to a stable environment, or what we may need to do to improve an environment to become more stable.